Hello, everyone, and welcome back. It's great to be here once again with all of you, and thank you so much for joining our webinar tonight. Today's topic is definitely an interesting one, definitely a bit of a difficult one, but also this is a topic we've never done before, so I'm very, very excited to simply have our new experts tonight with us. So thank you so much for joining. As you can see, there are two special guests tonight, so I'm very pleased to introduce you to Professor Soumeya Bekri and Dr. Abdellah Tebani, and they are both from Metabolic Biochemistry Department at Rouen University Hospital, which is located in France. And of course, it's great to have you here. How are you feeling tonight? Hope all is good.
Thank you very much. Thank you for your introduction, and thank you for your invitation. Thank you, Caroline, and thank you to the European Fertility Society for giving us this opportunity to share with you some thoughts.
Thank you so much indeed.
Thank you for a kind introduction. We're looking forward to share with you and to discuss about this inherited metabolic diseases and to share our experience.
Wonderful. Thank you so much. I'm really looking forward to it. Of course, Abdellah and Professor Bekri, they prepared a presentation for you, and they will explain everything. You will see that they will explain it in a very easy way, so don't worry. And I'm sure it's going to be interesting tonight for you. And of course, as always, after the presentation, it will be time for your questions. Go ahead, type those in the chat section. I'm sure they will be more than happy to answer any of your upcoming questions. And I guess we can go ahead, right, with the presentation. But before that, if you can say a few words about yourselves, that'd be great. Thank you so much.
Yeah, then I'm Soumeya Bekri. I am the head of the Metabolic Biochemistry Department. We are dealing with the diagnosis and follow-up of inherited metabolic disease. And the topic today is just to explain in a simple way this topic and the tools that we are using to do the diagnosis and the follow-up for our patients. And we will give you an overview of the metabolic landscape. And then after, we will just have a few examples from our experience in diagnosing some patients, different presentations. Then I will let Abdellah first, giving you the first insights, and then after I will give you some examples.
So I'm Abdellah Tebani. I work with Soumeya in Laboratory of Metabolic Diseases. So on daily basis, what we do is to try to diagnose and follow up our patients that have these kind of conditions. So the idea of this topic, as Soumeya mentioned in her introduction, is to give you some very, very simple basics of what these diseases are and how they occur and how we diagnose them. And we, in the second part, we're going to illustrate with real examples how do we diagnose them and to which extent these are important, how to follow them. And then we'll be happy to answer all your questions as well. So the topic today is about inherited metabolic diseases using some tools that are called omics. So the idea here is to give you some basic definitions of inherited metabolic diseases and how we diagnose them. So as you can imagine, a human being is just an assembly of different small parts that we call molecules. And these molecules, they come together to form very small structures to form at the end a human being. So the way a human being works is defined by how these small parts interact between each other through the full lifespan. So what is important to understand in any disease is how these different parts work together through the lifespan. So to be very in simple way, how do metabolism work actually? Because these diseases deal with metabolism. What is metabolism? Basically, is when you eat any food, it's broken down into small pieces. These small pieces are broken down through what is called metabolism. So metabolism is just converting the food, the big part, into tiny parts so that the human body can assimilate them. The final point of this assimilation is just to produce energy so that we grow, we speak, we feel, we see, etc. So this is basically what metabolism is all about. So these, to understand these diseases, we need to understand how this metabolism works to produce this energy so that we still alive and we live and we live basically. So when we break this metabolism into a very unique unit, it just goes as simple as this. We take a very small part that is converted into another small molecule called B. So we take the A part and it's converted to B through using a tool that is inside our body, which is called enzyme. So this enzyme 1 converts this small molecule A to B. Then another enzyme, another tool called enzyme 2 converts the product B into product C. So as you can see here, there is a highway that is drawn here from A to C thanks to these two small machines called enzymes 1 and 2. So these enzymes are proteins. These proteins, as you know, they come from something called the gene, which is in our DNA. So this metabolism basically is defined through our DNA. So this is the smallest part of our metabolism. So if you take a lot of pathways like this one, you will get something very complicated like this. So our whole metabolism is just an interaction between these small parts to form this whole metabolism, which is called a metabolic network. So what is a metabolic disease? As I explained earlier, to convert the molecule A to B, you need a machine. So this machine here is enzyme 1. So if your machine is broken, it's out of chart, it doesn't work at all, then you will get a lack of conversion from A to B. So you will get a lot of A that is stored because it's not converted to B, and you will get a lack of C because it's not converted from A to C. So in your body, you will get a lot of A and very, very low concentration of C. But since your body needs C to survive, or to C to talk, to produce energy, you will lack C. So then you will lack a function because you no more have C. In the other side, your body needs to get rid of A if it's normal. So if you have a problem in this machine, you will get a lot of A, and if A is toxic, then your body is in trouble. So this is the first part to understand what is a metabolic disease. What I said earlier is that this machine is actually coded in your DNA. with your gene 1 that codes for machine enzyme 1, gene 2 that codes for enzyme 2. So if you have an alteration in your DNA, in your genome, then you will get a problem on your machine enzyme 1. And if you have a problem in your DNA, that makes it inherited because you transfer it through generation. That's why we call this disease inherited metabolic diseases. Inherited because they are carried on genes, metabolic diseases because they are due to an impairment in your metabolism. So that's what a metabolic disease is. So if you take the whole metabolism, almost 1,000 diseases have been described so far that causes a metabolic disease because one of these 13,000 machinery is altered. So why inherited metabolic diseases are important? Inherited metabolic diseases can appear at any stage of your lifespan. They can appear in prenatal stage, so in the womb. They can appear in perinatal stage when you are very, very, very little, in neonatal stage, and you have a lot of other diseases that span through the whole lifespan. So inherited metabolic diseases, it's not a pediatric disease only, but it spans through the whole life. So why we should diagnose inherited metabolic disease? Thankfully, when we understand inherited metabolic disease at the basic level, we can act through different therapies. So if you take the basic line pathway that we explained earlier, you have A that is converted to B, that is converted to C, thanks to enzyme 1 and 2. In this case, our machine that has the problem is enzyme 2. So if you have a problem on machine 2, as I said, you have an increase of B and decrease of C. And what makes the disease is that you have a lot of B, but also because you have low concentration of C. So how could we treat these diseases? So either you restore the B and you get rid of the increase of B so that you have no more toxic effects. So you try to get rid of B by activating another pathway. So you just open another door so that the B goes through so that you don't have that toxic effect. This is the first part. The second part that you can use is you just give your patient the molecule that is lacking. Here is C. So you can either give directly C or give some other drug that can produce C so that you will increase the concentration of C, and then the patient is no more suffering from the disease because he gets or she gets the lacking molecule through drug. The last part, what you can do, if you have something that's broken in your car, you can just replace it with using a spare piece. So here is the same thing. We have a problem on this piece, which is enzyme 2. So what we can do is just replacing it through injection. We just bring enzymes and we inject the patients with the new enzyme, and this is called enzyme replacement therapy. So you have 3 ways to treat a metabolic disease. Either getting rid of the toxic metabolite or molecule. 2, replacing the important metabolite that is lacking, and third is replacing the broken machinery, which is the enzyme, through injection. So these are the main therapeutic weapons. Now, how do we diagnose these diseases? What are the tools? What is the way we do this? So let's get the basic that we're talking from the beginning. You have a molecule A that is converted to B through an enzyme or a protein that is produced by a gene. So the first thing to do to diagnose a disease is to prove that you have an increase of A or a decrease of B. And we do this using a technology called metabolomics. So we take samples from the patients, either urines or blood, often, and we should see if there is an increase of metabolite 1 or a decrease of metabolite 2. So this is the first step. The second step is to prove that this enzyme 1, so the machine, doesn't work anymore or doesn't work enough anymore. So the way we do this, we use what is called proteomics, basically. The third tool that helps us to diagnose inherited metabolic diseases is to prove that there is a problem in the DNA that produces your machine. And that is done by interrogating the gene structure. So then we use a tool called genomics to prove that there is a variation, a pathologic variation, a change in your DNA sequence that produce a wrong machine that doesn't work. So basically, we have three tools to diagnose an inherited metabolic disease. First, we have to prove that there is an increase or a decrease of a metabolite that is related to that disease. Second tool is to prove that your machine doesn't work anymore or doesn't work enough anymore. Third tool, what is very important, is to prove that this disease is inherited. So we need to prove that there is a change, a problem on the DNA. And for this, we use genomics. So we have three tools: metabolomics, proteomics, and genomics, to be able to diagnose inherited metabolic disease. So now, since we have these different omic tools, so far, inherited metabolic diseases are not seen in that simple way. We need to put the patient in his or her environment. So the patient evolve in a social, environmental ecosystem. So to be able to precisely diagnose the disease and go through a serious follow-up of the disease, we need to know where the patient lives, how he eats, which other diseases he would have, what is his diet, does he exercise, etc. And then we need a very precise picture of what he feels physically, so on the clinic side. So we have three parts to be able to precisely diagnose inherited metabolic disease. We need to prove the biological problem, we need to shape the ecosystem in which our patients evolve so that he gets the presentation we see physically. So now Soumeya will give you some real life example before going to the Q&A sessions.
Then, as Abdellah said, we have all the possibilities for the onset of an inherited metabolic disease, from the prenatal state to the elderly state. Then if I focus in the first part on the prenatal part, presentation, we can have a prenatal presentation of inherited metabolic disease during the pregnancy. And we know that for a long time right now. And the main entrance, the main sign is hydrops fetalis. Hydrops fetalis is an edema of the fetus. And we know some really common etiologies like immune etiology. But if we read of these commune etiologies, we have a lot of hydrops fetalis without any diagnosis. And we know that inherited metabolic diseases came with this presentation, but with conventional biochemical investigation, we are not able to do all the diagnosis, because we are doing some analysis in a sequential fashion, and we have an hypothesis. We think about a disease, then we will try to explore this disease, and if it's not, we will have another hypothesis, and that so on. Then we ended up with having a lot of cases without any causes, known causes, and we cannot handle these patients. Then we, in our lab, 5 or 6 years ago, we decided to perform a new generation sequencing analysis, including all the known genes able to cause such a condition, such prenatal presentation of inherited metabolic diseases. Then in this way, we can analyze 42 pathologies in one shot, and we can have a data-driven hypothesis, because we are looking for a global analysis, and we end up in targeting one pathology. Then here we are doing a genetic analysis at the first line as a first line investigation. Then when we end up with a pathology, we can confirm using the other way, as Abdellah said, we have proteomics and metabolomics study. Then we can do all this biochemical confirmation afterward. Just to give you an example, we had a female patient, pregnant, 35-year-old woman. At the prenatal ultrasound examination at 31 weeks of gestation, the clinician, the examination showed a kidney abnormality with the high size of kidney, and we have performed different genetic, viral, metabolic etiology and with investigation, and we don't have any clue for this condition. And when we perform this NGS analysis with the hydrops fetalis genes, we end up to identify an alteration of a gene that is known to cause kidney disease. Then we the end of the pregnancy went well, and we had an uneventful birth, and we were able to provide an early management of the renal function for the baby, and he's doing well with the normal psychomotor development right now. And we were able to do also genetic counseling afterward. Then this is just an example for the prenatal. The majority of inherited metabolic disease have a neonatal presentation. This is an example. We have often some days or some hours or some days without any symptoms free. And here it was a little girl, and she presented at 12 hours of life and anorexia, poor consciousness, hypothonia, and abnormal movements. And all these clinical signs help us to think about metabolic impairment. And we have done some metabolic analysis, and we end up by demonstrating that we have some abnormal amino acid concentrations with an elevation, as Abdellah said, metabolite elevation and some decrease in other metabolite that allow us to determine the protein, the enzyme that is not working. And in this case, it was a disease that we know very well in our practice. It's maple syrup urine diseases. And the good news is, even if it's a serious disease, the good news is we can manage it using an appropriate diet. And the prognosis is changed completely if we are able to done the diagnosis in very early stages of the disease. Then we can have almost a normal life, and we were able in this case to perform genetic counseling as well. And to give an example of a late presentation, because in the medical community, inherited metabolic diseases are always linked to pediatrics, and people are not aware that we can have late presentation. Here we have a 45-year-old women with serious liver abnormalities, ending up in fibrosis and steatosis, then fibrosis. And when we assess the lipids in blood, we showed that we have an increased level. Then for the diagnosis, we have done first metabolic analysis, then we assess the enzyme activity of an enzyme in lipase acid enzyme, and we have this decrease. Then we confirmed the diagnosis by using genetic analysis, and the disease is cholesterol ester storage diseases. This disease is treatable by enzyme replacement therapy, and we can also perform genetic counseling. Then I will let Abdellah concluding this talk.
So in the end, what we wanted to show with this very short talk is that to show there is a hope in increasing the way we diagnose this disease, and of course treating them, because now, as the title of our talk showed it, is that now we have new lenses to look at these diseases at unprecedented depth and resolution. These new tools basically next generation sequencing, mass spectrometry, and other tools all these tools allow us to dig into a more detailed visualization of these problems at the metabolic level. So when you see things at this depth, you can understand them better, you can treat them better, and you can counsel the patients better. So the next generation of healthcare in this disease and in healthcare in general will be heavily based on these different omics technologies, and all you hear in the media and about artificial intelligence and this big data thing, this will help us a lot to provide better healthcare to the patients, because at the end, what we all seek is more precise information, more precise treatment, and healthier life when we do understand things at a higher resolution. So the way we do diagnosis now, it's steering towards a new way by relying heavily on these omics technologies that allow us to see things we were not able to see before. So now I would just want to thank all, and there are a lot of people working with us to handle these diseases. There are few only, and of course we can't thank more patients, because they are the heroes of all these stories. So thank you very much for listening and for your kind invitation. So we'll be happy to answer any questions if you have.
Amazing. Thank you so much. You have brilliantly explained this. I think it's now a little bit more clear to anyone that was not that familiar with the topic. And it's definitely interesting to know that there are some options to deal with it. So incredible job, that's for sure. Thank you so much. And now it is time for your questions. So as always, type those in the chat section. There are some questions ready. And okay, let's get to them, okay? Ready?
Ready, go ahead.
Okay, so the first one is right here. Do these presentations of women have had any male factor?
The presentation for inherited metabolic disease, the main transmission is equal for men and women. We can have the same clinical signs in men and women, except for the disease that are transmitted through the chromosome X. Then for the chromosome X, we have, for example, Fabry disease. It's a rare disease, and the gene is on the chromosome X. Then the women are more protected since we have two X. That in this case, we can have some difference between the presentation between male and female. For the male, since we have only one X in the male patient, then if the only one copy of the gene is altered, the disease will be present and will appear, and all the symptoms will be present. But in the female, it depends, because it's quite kind of complicated, but we can have more moderate presentation in female, or sometimes in some disease we can have no presentation at all. But this is the difference. But for the majority, 99% of the inherited metabolic diseases, we have the same presentation for in male and female. I hope that I answered the question, but the example that we have here, it was female, but it's just by chance.
Yeah.
Okay. All right. Thank you so much. And of course, if you have any follow-ups, you know what to do. And actually, yeah, thank you. Okay. Thank you for joining still, of course, Derek. Thank you so much. And let's have a look, okay? If you can provide some examples of inherited metabolic diseases as well.
Yes, in the majority in Europe, at least, we have in all our countries a program of newborn screening. I think that you are aware that for each newborn in Europe, at least, it's not the case in some other countries such Africa or other countries, that poor countries, but in Europe and Occidental countries, we have this newborn screening, and in this program, it's national program, systematic screening that help to detect and to diagnose and to screen for inherited metabolic diseases that are treatable if the diagnosis is done very early. That's why we decided start from the beginning of 1917, something like that. Then it's a systematic screening to detect these diseases and to provide with the diet adapted, appropriate. Generally, it's diet, but sometimes it's treatment. But in general, it's diet, just to have this block, this metabolic block, bypassed using another pathway, metabolic pathway. And just to give you an example, I think that you heard about phenylketonuria or PKU, that this disease is part of all the newborn screening in Europe.
And no, worldwide as well. Most of
This is, but we can have inherited metabolic diseases. We can have some, at least examples in the three major pathways: sugar, lipid, and proteins, and all the enzymatic reaction, the enzymatic pathways can be targeted, can be blocked. Then we have all the huge network that Abdellah showed you can be broken at a step.
All right. Yeah. Thank you so much for explaining that question once again as well. Okay, let's have a look. Okay, the previous question we got from Kate. Is it true that women with PCOS are more likely to have metabolic syndrome? Do you also help such patients?
What do you mean by PCOS?
Polycystic ovary syndrome okay. Condition. Yeah.
Yeah.
It's not inherited metabolic diseases.
Yeah, it's something else.
Yeah, it's not inherited metabolic polykystosome.
And
yeah, it's not inherited metabolic diseases. But this question is very interesting because we are talking about genetic diseases, inherited metabolic diseases, and here we are linking a genetic disease with a metabolic syndrome. And now we know that monogenic disease don't exist, because even if we have only one gene that is not working, we have all the environment that can have an influence on this disease. And it's more complicated than one gene equal one disease. And now we can see that for some genetic disease, we can have other disturbances of the metabolic network. And since now we have more tools, omics tools, for instance, we are able to have a wider view of the network, and we can see some consequences that we are not able to see before.
All right. Thank you so much, Kate, of course, for that question and explaining that as well. And so for how long is Omix available, if I can ask that additional question as well?
different tools, actually, different omics tools, we do use them on routine basis, actually. But those that are really being used in the clinic, it's mainly next generation sequencing, which is genomics, which is heavily used in the diagnosis of genetic diseases, metabolomics, because we look at metabolites, and proteomics as well. It's getting more and more involved, and the prices are getting very low now, and most of the labs can afford these genetic diseases at affordable prices, actually.
All right. Thank you so much for that answering that additional question. And let's have a look. Okay, more questions are coming in. How would you know when to test for metabolic disease? Are there any symptoms to look for that could be ignored or misunderstood as something else? Thank you for the question.
There is a lot of science that can help us to think about an inherited metabolic disease. For pediatrician, it's straightforward. They are used to, and the main problem is the shortage of energy. If you have not enough energy, we have some organs that cannot be well. The first one is the brain. Then the first sign, for the children at least, the first sign is coma or neurological decompensation, epilepsy, or so on. Then in pediatric stages, we don't have concerns, because pediatricians are aware, and they are if they see that the metabolic disease can be suspected, they do what they have to do very quickly, and we can do the diagnosis and take care of the child very quickly. But since we told you, we insist on the fact that the symptoms can appear anytime. For adult patients, we can have some delay in the diagnosis, because the medical doctor dealing with adult patients are not really used to this kind of disease. Then sometimes we have patients that have very symptoms since several years, and the diagnosis is very, very, very late. Then that's why I think that we have to continue to talk about these diseases, and to
raise awareness.
Yeah, to raise awareness. Yeah.
May I add something about this? One of the some symptoms that we usually see in this kind of disease is that, for example, it's not always the case, but just to give you an example which is obvious, some kids, for example, when you give them a specific diet or a food, for example, in some cases when you give some meat to a kid and he has some reaction about that, we can't think about something, some specific metabolic disease, because we can have the link about high protein diet that could cause some comas. In some cases, we can think about this way. But as Soumeya said, one of the challenges of getting this metabolic disease, because they are called, and we don't agree about that, but they are called rare diseases. They are not that rare, because if taken as a group of disease, they are more often than we could imagine. So one of the challenges we all face, and I think this kind of meeting try to fill in the gaps, is to raise awareness of these so-called rare diseases, because they are not rare, as I said. So sometimes, just because you know that this could be a metabolic disease, and it just light a ball in your head, and you can go through the right pathway. And sometimes it's just a matter of hours or days, and you can save a lifetime of a kid. So it's not straightforward, but just thinking about it is a way to go.
Thank you so much indeed. Yes, that's one of the reasons we are here, to just spread this information so anyone will be able to just get familiar a little bit more. Yeah, so thank you so much indeed for that explanation as well. Let's have a look. Okay, more questions are coming in. You kind of answered this already, but you can answer to Kate. Are the rates of metabolic syndrome the same in men and women?
Yes.
Yeah, roughly. Yeah, it's roughly
yes.
it's the same.
Okay, thank you for the clarification once more. And would siblings always have the same metabolic disease if it is present in the parents' genes?
It depends. It depends the way the disease is transmitted.
This question is similar to the previous one, because we are not, if we have a monogenic disease, we can have with the same gene, with the same alteration, with exactly the same context, the sibling, we can have, and we had last week, we had the case in our lab, with a case with the moderate presentation, and in the family, we have exactly with the same variant, the same alteration in the gene, a very severe presentation. And that's why we think that other factors, genetics factors or environmental factors or whatever, we are not able for all the diseases to have the exact answer right now, but for sure we have this heterogeneity in the phenotype, in clinical phenotype, and we are not able to say that this disease will behave exactly in the same way for all the siblings in this family, for example. Even if sometimes we have some gene alteration that are severe and others that are moderate, but even we have this individual specificity. And that's why the omics tools that we are talking about enable us to have a huge amount of data that can represent this heterogeneity, that can enable us to approach this complexity.
Actually, I just want to add something about this. The way we presented the disease so far, and we did it, of course, just for sake of simplification of the talk, this linear vision about a disease, that if you have A, you have B, and it's linear if another sibling has exactly the same thing. Biology is not at all a linear phenomenon. And in our research, actually in the lab, we are using omics tools and other tools just to prove that unfolding this disease complexity is for sure not linear. That's why what we said in the beginning is that calling these diseases monogenetic diseases is not that true, because as we said last week, we got the same gene alteration, totally different clinical presentation. So this obviously and eloquently show that biology is not at all linear.
Once again, thank you for the thorough answer to yet another question. And let's have a look at the next question. Any thoughts on mitochondrial injection that are autologous?
For the mitochondrial diseases, the question is quite different. The transmission might be different. Mitochondria is an organelle. It's a small part in the cells. And this, in the mitochondria, we have a small DNA that is different from the DNA that is in the nucleus. And this small DNA encoded for only 13 proteins. All the other proteins that are needed for the mitochondrial functions are encoded by the nuclear DNA. Then we can, the point is that the mitochondria is inherited only from the mother, from the ovule. Then the inheritance is maternal. Then if a woman have a mitochondrial disease due to the alteration of the mitochondrial DNA, she may have children with this disease, male and female children. But the male with this kind of mitochondrial disease will not be able to transmit this disease to his children. Then it's easier to predict and to counsel if we have a mitochondrial disease with a nuclear origin, because it's straightforward. We can have a variant in the fetus, for example, for a prenatal diagnosis, or not, and we can counsel based on this. For the mitochondrial transmission, if the variant is on the mitochondrial DNA, it's more difficult to be sure that this mitochondria, for the maternal mitochondria, is not inherited in the fetus or not. I don't know if it's enough clear, because it's kind of complicated, but this is the
because mitochondrial diseases, it's not straightforward transmission, so it needs some schemes just to show how things work. But Soumeya answered, I think.
Thank you so much. And actually, there's a thank you from Tarek for you as well. Thank you indeed. And it looks like that will be our final question now. But of course, if you have any left, do it now. It will be final call for the questions. And the question is easy, I think. Is metabolic syndrome reversible?
Not the metabolic.
Metabolic syndrome, even if it's the name is metabolic syndrome, it's not an inherited metabolic disease. It's a metabolic impairment, but it's an acquired metabolic impairment. It's not inherited.
Yeah.
But to answer the question as well, it depends. It depends at what stage the diagnosis is made, and what the
age.
What age and what stage, and all the environment and the diet that is taken by the patient. But yes, in some cases it's reversible.
I just, if I might add just one thing about this. If you look at the slide I showed about environment, biology, and clinic phenome, and I think metabolic syndrome fits very well in that picture, because it depends in the 3 layers. You have the 3 layers, and one of the challenges to embody the precision medicine perspective is metabolic syndrome as well, because it takes the biological layer, the environmental layer, and the clinical layer.
Okay, that makes perfect sense, of course. And thank you for the clarification. Yes, so thank you. It's been definitely interesting. I'm just checking. Someone is typing. Let's give it a second. Let's see if it's another thank you or a question. But of course, you know, if you haven't asked anything tonight and you would like to get some more details, you know, you will be able to get in touch with Professor Bekri and of course Dr. Tebani, sorry. And of course, I'm sure they will be more than happy to help you out. After we finish, you will be redirected to the website, and there is a contact option. And of course, those messages are always sent to you, so you will be able to definitely answer, I'm sure. And of course, we will be finishing for tonight, but thank you so much. This has been very, very interesting, but I think that, well, I will show you a comment that will make sense what I wanted to say. So helpful. I have never heard of this type of diseases before. And I think lots and lots of people feel very, very similarly. So thank you so much for bringing this for sure. And as you can see, thank you.
Thank you.
Thank you for you.
We were very, very pleased to spend this moment with you.
We are happy that you are here with us, that's for sure. And is there anything else you would like to add?
Yeah, I really like to thank the way this event is organized, and that the audience is very interacting and very, very precise and thoughtful questions, sometimes challenging, but that's a very interesting talk.
That's great to hear. And of course, yes, what can I say? Thank you so much for your questions. That's for sure, because of course we are here for you, so to help you in any way we can. And I'm sure it has been useful and interesting for sure. And just so you know, of course, this has been recorded. You will have a chance to watch this again. You will find it on my IVF Answers, but also on our YouTube channel. So I can encourage you only to subscribe. That way you will know when the video is uploaded. And again, thank you so much, Abdellah, Soumeya. It was amazing to have you here. Thank you once more. And of course, now everyone have a relaxing weekend. Of course, we will be back on Monday at 8 p.m. UK time. There is another webinar and another topic on genetics. So have a look and sign up already. And thank you to, of course, for joining us. And again, it's been great to have you here.
Thank you.
Bye-bye.
Thank you. Take care. Take care. Bye.